Dear viewers, welcome to Food Microbiology. In this session, you will know about microbial growth in foods. Microbial growth refers to increase in cell number, but not in size of the cells. In bacteria, growth is always followed by division of the cells by fission, leading to increase in cell number. This increases the population size. Accordingly, there are more number of bacterial cells than cells larger in size. Growth rate in microorganisms is very high and microorganisms multiply very fast in a suitable environment. Under favorable conditions, microorganisms multiply rapidly and maintain their small size. However, there is no change in their morphology. This unit is covered in 6 sections. They are growth in bacteria, bacterial growth curve, diaxic growth, synchronous cultures, microbial growth in food, bacteria, yeast and molds. Factors affecting microbial growth in food. Now, growth in bacteria. Bacteria multiply by binary fission. A single bacterial cell divides into two after the development of a transverse cell wall between them. Many bacteria can divide in less than 15 to 20 minutes under favorable conditions. During the cell division, complex biochemical changes takes place. These are synthesis of polymers, subcellular components and other required materials. Several events takes place before cell division as shown in the figure. Nutrients from surrounding environment are transported into the cells. These nutrients are converted into cellular components and enzymes, etc. The nuclear content increases, which is sufficient for both the cells. Elongation of the cell takes place. The cell contents are distributed evenly. The cytoplasmic membrane invaginates at the center of the elongated cell. A transverse cell wall is formed, presumably where mesosomes are present, leading to separation of two cells. Thus, one cell becomes two in a very short period. If cell division occurs in bacteria which are in chain form, for example streptococci, these cells remain together. If the next cell division takes place at right angles to the plane of the first division, it appears as tetrad, for example pediococcus species. If division occurs in their plane, it results in a group of 8 cells, for example Sarcina species. If cell division takes place in different planes, it may result in a bunch of cells like in the case of Staphylococcus as shown in the figure. Growth rate and generation time of bacterial growth. Most bacteria divide by binary fission and the number increases in geometric proportion, like one cell becomes 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64 and 64 to 128, or this can be expressed as 2 to the power of n, where n is the number of generations. Generation time. The time required for the bacterial cells to double its population is known as generation time. It differs based on the organism and the growth conditions. For some bacteria, it may be few minutes, or for some other, it may be several hours as shown in the table. It depends on the type and the amount of nutrients and the environmental conditions like temperature, pH, amount of oxygen present, the presence of inhibitory compounds, etc. The growth rate of any bacterial population can be calculated if the number of generations denoted by smaller n in a given time is known. The bacterial growth rate can be expressed in terms of mathematical expression. For this, the initial bacterial number denoted by capital B, the number of bacterial cells at the end of the given time denoted by lowercase b, and the time duration are required. The number of generations can be determined by using the formula n is equal to 3.301 into log lowercase b by capital B. The generation time G is the time interval required for the cell population to double. It is determined by dividing the duration of time t by the number of generations n, that is generation time is equal to t by n. Bacterial growth curve. It is a graphical representation of various growth phases of a bacterial population. To understand this, an experiment is conducted by inoculating a small number of bacterial cells into a growth medium. Cell number is estimated in terms of viable count after regular intervals. The viable count is plotted in log numbers against time period. Alternatively, cell count is estimated by colorimetry or by direct microscopic methods. However, in these methods, dead cells also contribute to the turbidity. Microscopic observation or turbidity cannot differentiate live and dead cells. If the bacterial growth curve is plotted by using the data collected by colorimetry, the decline phase of the growth curve cannot be seen, as decreased absorbance is not depicted. A typical bacterial growth curve has 4 major phases, and there are 3 transitional phases separating these phases as shown in the figure. Lag phase. It is an initial growth phase and no visible increase in cell number in this phase is noted. Cell enlargement takes place along with the synthesis of new cellular components, DNA, cell organelles, enzymes, proteins, etc. Cells may need time to adjust to the new physical or chemical environment. During this phase of adjustment, synthesis of additional cellular components for the new cell takes place. Cells are active with increased quantity of proteins, RNA, DNA and other components. The duration of this phase varies with the type of bacteria and other factors. Dormant cells may take long time. If water activity is reduced in a food, duration of this phase can be extended. There is a lag in cell division. All the cells do not complete this phase at same time. Gradual increase in cell number is attained by the end of lag phase, hence a slow upward increase in the graph is seen. The rate of multiplication increases with the time and reaches the maximum at the end of this phase. Cells are sensitive to unfavorable conditions. Log phase. This phase is also known as exponential growth phase. In this phase, cells divide steadily and at a constant rate. This results in a straight line in the graph when drawn against time. Cells are smaller in size and active during this phase. The entire population is active in this phase. Hence, cells from this phase are preferred to be used as inoculum for metabolite production. During this phase, generation time is very short and constant. The growth rate is maximum and constant. However, at the end of this phase, decline in the multiplication rate results in a decline in the graph curve. This is due to the accumulation of toxic metabolites like acids and change in the environment of the medium. Duration of the exponential phase may vary depending on type of bacteria, amount of nutrients and environmental conditions. Stationary phase. In this phase, the number of new cells formed is equal to the number of cells dead. As a result, the growth curve appears as flat during this phase. However, it turns downwards and reaches maximum at the end of this phase due to increased death rate. Death phase. In this phase, the number of viable cells decreases exponentially, resulting in the graph to move downwards. If the initial population at the beginning of this phase is 1 million, it becomes half million, 1/4 million and so on with the increase in the incubation period. In this phase, death rate is constant and varies from bacteria to bacteria. Depletion of nutrients and accumulation of toxic components are the main reasons for death phase. Gram-negative bacteria die faster than gram-positives. Spore formers can survive long period. Diaxic growth. It is the growth of a bacterial population wherein growth can be seen in 2 separate phases. This is due to the preferential use of one carbon over the other. If the results of diaxic growth are plotted, a graph as shown in the figure can be obtained. This has 2 exponential growth phases separated by one transient lag phase. This is due to the catabolic repression of induced enzyme synthesis. This feature indicates that bacteria try to survive in the environment by making use of all possible resources available to it. For example, if the cells growing in xylose containing medium are added to a medium containing both glucose and xylose as carbon sources to grow, bacterial cells first use xylose for its metabolic activities and later it tries to survive by using glucose, which is already present in the medium. However, cells require some time to change its metabolic activities to use a hexose sugar by synthesizing the enzymes required to metabolize the glucose. Hence, a lag phase can be seen in between two log phases. Synchronous cultures. In a growing culture, bacterial cells have different stages of the growth cycle. Some may be very young, some may be well developed, and some other about to die. It is very difficult to predict the behavior of the population in its condition. Hence, there is a requirement of having all cells at the same stage of the growth. This can provide behavioral information related to the whole population. A synchronous culture is thus defined as a population of bacteria that contains cells that are all in the same growth stage. A population of bacteria can be synchronized by manipulating the physical or environmental conditions. When bacterial cells are inoculated into a fresh medium and incubated at a less than optimum temperature for around 10 to 12 hours, bacteria start assimilating nutrients from the medium. However, cell division will not take place at this temperature. Once the medium is transferred to optimum temperature, all bacterial cells start dividing at the same time, leading to synchronous growth. Filtration is another method to get synchronous cultures as shown in the figure. The bacterial cell population grown is filtered through a filter which can retain bacteria over it. Later, this is inverted and fresh medium is allowed to pass through it. All bacterial cells adhered to the filter start dividing simultaneously, leading to the synchronous cultures. However, this can be maintained for a few generations only. Microbial growth in food, bacteria, yeast and molds. Food is a rich medium for the growth of microorganisms. It contains carbohydrates, proteins, fats, minerals, etc., which can support the growth of microorganisms. Some microbes can grow in the foods and cause health hazards to the consumers. For example, Salmonella, Staphylococcus aureus, Bacillus cereus, E. coli, Listeria monocytogenes, Clostridium species, etc. The growth of some other microbes may lead to spoilage of foods like several molds, yeast and bacteria. These microbes may bring enzymatic changes, produce off flavors and change the color. All this happens due to the metabolizing of various components present in the food. This makes the food unfit for consumption. Some other microbes, for example, lactic acid bacteria, Saccharomyces, Aspergillus oryzae, etc., can convert the food from one form to another, leading to development of several fermented foods. As discussed earlier, the generation time may be as short as few minutes for several microbes. Hence, foods may get spoiled rapidly if it is not protected. However, several factors can influence the growth of these microorganisms. Factors affecting microbial growth in food. Several factors affect the growth of microorganisms and their metabolic activity in foods, changing the characteristics of the food itself. These changes may be considered as beneficial if it happens during fermentation, leading to production of fermented foods like curd or cheeses. When the changes lead to spoilage of foods or causes pathogenicity to the consumers, then they are considered as harmful. Factors affecting the growth of microbes are intrinsic factors, extrinsic factors, implicit factors, processing factors, and interaction among these factors. Intrinsic factors are factors pertaining to the food itself. For example, pH, water activity, redox potential, nutrient content, presence of inhibitory compounds and mechanical barriers, etc. Extrinsic factors are factors associated to the environment in which the food is stored. For example, relative humidity, temperature and gas composition, etc. Implicit factors are factors associated to the microorganisms themselves. For example, interactions among different microorganisms growing in the food and between the food and the microorganisms, ability to tolerate various stress conditions in the food, production of antimicrobial compounds like bacteriocin and growth promoting compounds. Processing factors are factors related to the processing of the foods like freezing, cooling, drying, heating, etc. Interaction among these factors in combination may either promote or decrease the growth of microorganisms. Intrinsic factors. Several intrinsic factors can influence the growth of microorganisms. The pH can affect the growth of microorganisms. Based on the pH requirements, bacteria are classified as acidophiles, neutrophiles and alkalophiles. Many bacteria can grow better in the pH range of 6 to 8, yeast between pH 4 and 6 and fungi between 4 and 10. Many lactic acid bacteria have the ability to grow in acidic range of foods. Change in the pH affects the enzyme activity, permeability of membranes, solubility of cellular constituents and shifts the metabolic activity. Water activity represented by AW is referred as the ratio of vapor pressure of water above the material P to the vapor pressure of the pure water P0. It is an index of available water in the foods. As a solvent, water is an essential requirement to transport nutrients and waste, hydrolysis of proteins, fats, polysaccharides, etc. If the water activity of the food is less than optimum for the growth of microorganisms, the lag phase of microbes extends by several hours. Hence, foods can be preserved for longer period if the water activity is reduced or controlled. Water activity in foods can be reduced by adding sugar, salt or by drying. The natural antimicrobial compounds like short chain fatty acids, essential oils or natural antimicrobial compounds like lysozyme in eggs, lactoperoxidase in milk can inhibit the growth of microorganisms. Some of the antimicrobial compounds may be bacteriostatic, which inhibit the growth by stopping the division, and some bactericidal, that is killing the bacterial cells. Depending upon the compound, membrane damage, inactivation of enzymes or nuclear material can occur. Some may cause mutations by alkylation or deamination. Extrinsic factors. Several external factors can influence the growth of microorganisms. Relative humidity RH is the amount of free moisture present in the air. It is an essential measure of the water activity of the gas phase. Water activity and relative humidity are interrelated. If any food with low water activity is kept in a place having high RH, water will enter the food through gas phase. This will finally affect the quality and acceptability of the foods. Temperature. Temperature plays a major role in the growth of microorganisms. Based on the temperature requirement, microorganisms are divided into three groups: psychrophilic bacteria, mesophilic bacteria and thermophilic bacteria. Besides this, some of the mesophiles can also grow well at low temperatures. For example, Listeria species. Higher temperatures can inhibit the growth or kill the microorganisms due to the disruption of cytoplasmic membrane, enzyme inactivation and denaturation of proteins and DNA, etc. Oxygen. Based on oxygen requirement, bacteria can be grouped into obligate aerobes, obligate anaerobes, facultative anaerobes, microaerophilic and aerotolerant anaerobes. Obligate aerobes cannot grow without oxygen, for example, bacillus. Obligate anaerobes cannot grow in the presence of oxygen, example, Clostridium. Facultative anaerobes can grow with or without oxygen, for example, Staphylococcus E. coli. Microaerophiles require less concentration of oxygen, for example, lactic acid bacteria. Aerotolerant anaerobes do not require oxygen, but can tolerate oxygen at certain concentration, for example, Enterococcus. Superoxide dismutase, catalase and peroxidase are required to inactivate the free radicals like superoxide, singlet oxygen, hydroxyl and peroxide anion generated by oxygen. As anaerobic microorganisms do not produce these enzymes, they cannot survive in the presence of oxygen. Processing factors. The food processing methods like freezing, cooling, drying and heating etc. hamper the growth of microorganisms. These methods either remove free water from the cells or cause damage to cell membranes. Microorganisms can inhibit or stimulate the growth of each other. Microorganisms are always present in mixed groups. The goal is self-preservation, hence they try to compete for food. The resulting interaction may be either beneficial or harmful. For example, Saccharomyces produce alcohol, whereas film yeast, for example, Debromyces, breaks down this alcohol into carbon dioxide and water. In yogurt, symbiotic association between Streptococcus thermophilus and Lactobacillus bulgaricus can be seen. In the presence of micrococci in milk stimulates the growth of acid production by lactic acid cultures due to the production of catalase. The bacteriocins, for example, nisin, pidiocin produced by some lactic acid bacteria can inhibit the growth of pathogenic bacteria. In conclusion, we can say that bacteria multiply by binary fission. The generation time varies in the bacteria. While growing in a medium, bacterial cells appear in different phases of growth. These are lag phase, log phase, stationary phase and death phase. This indicates that all bacterial cells are not in the same phase of the growth. As the bacterial cells are too small in size, they do not have place to store many enzymes. To survive in the nature, bacteria show preference over the use of carbon sources. As a result, they take additional time, lag phase, to synthesize enzymes required to assimilate a new carbon source. To know the bacterial information related to the whole bacterial population, synchronous cultures wherein cells are in the same growth stage are required. Bacterial growth in foods is influenced by several internal, external processing parameters or combination of all these. Thank you.